Good morning, students. Welcome back to online class. Today we are going to do the biomolecules content, the last topic. So we are here in the proteins. So as you know, protein is important in our body. Almost 20% of protein is required in our body in a daily basis. Depends on the different weight. So the function of the protein is it is used for the growth, tissue repair, transport nutrients across cell membranes, that is a glucose transport into the cell, which is act as a intracellular ground substance. That is example is collagen. The name of the protein, there is a different types of protein is there, like keratin protein is there in our hair, then nails. So in different organs of our body. So act as antibodies to fight infection organisms. It is also act as a receptor, for example, receptor of smell, taste, hormones. So some are hormones, that is example insulin, enzymes, trypsin, pigments, that is example is hemoglobin. So these are all the examples of a protein, can be used in different ways in our body. So here that is amino acid, which is an important form of a protein. So amino acids in a form of glycine, alanine, and valine. So what is a glycine? The structure of a R. What is R? Is an alkyl. So alkyl can be changed in a form of H, CH3, and CH3 twice. That is the isoform of a valine. So the prefix can be written as a gly, ella, or val. We can symbolize in a form of capital G, capital A, and V. So understood. So this is all the form of amino acids, that is a glycine, alanine, and valine, as well as leucine. So CH2, CH3 twice. This is a tertiary form of alkyl. Isoleucine. Isoleucine means CH2, CH3. Capital letter is I. Phenylalanine. So phenyl means benzene group, benzyl group, or phenyl. Alanine means CH2. So PHE or capital F is written. Asparagine. The name you have heard for the medical form. So CH2, CONH2, amide group. CONH2 is amide group as well as CH2. So asparagine. Glutamine. So it's a glutamine, that is CH2, CONH2, GLN, or Q we can write serine. That is alcohol, form of alcohol, CH2OH, and the bond is attached with the CH2OH. Theorine, that is CHOH with the CH3. Cysteine, CH2SH. Cis means cysteine. Methionine, CH2SCH3, which is form of a sulfide. Triosine, benzene ring with OH and then CH2. Benzene ring with OH and CH2, that is triosine, TYR. Tryptophan, that is a formation of a benzene attached with the amide. There is a pentaform NH and then CH2. That is a trip form. It can be written as formula as a W, tryptophan. Proline, that is NH, COH and H. It's a pro form, proline, which is a combination of a amide as well as carboxylic acid. So proline, the formation is proline. Some more things is there. That is aspartic acid, CH2COOH. Glutamic acid, that is CH2COOH, glue form. And some basic amino acids, there is an amine form, that is CH2 times NH2, CH2 thrice, NH, C double bond NH, NH2, that is arginine. Histidine, that is a formation of a N with a pentane form NH and then CH2. This is a histidine. So these are all important 20 amino acids can be learned from. There is some other forms of amino acid, that is a D and L form. So how we can write D and L form? We have a dextro and liso. We have learned in the previous one. So D form with what? NH2 in the right side and NH2 in the left side is the amino acids. Fine. So accordingly, the carboxylic acid is there in the amino acids. Amino means what? Amine group, as well as acid means carboxylic acid group is there. That's why the name is given amino acid, which is used for the preparation of the proteins. Some other proteins is there. It is a globular protein. The structure is somewhat like this protein formation. So the example is insulin, albumins, which is present in the egg. So that's why it is rich in proteins. So 4 different types of protein, that is the primary, secondary, tertiary, and quaternary, is discovered in 1954 by the Linus Pauling. So we will study about the primary structure of the protein, which forms a disulfide link, that is SS bond link is there, which is present in the insulin in human. This is a secretion of insulin in human, which is important for the body. So if there is a lack of insulin in our body, then we can human can get chances of the diabetes. So that the insulin is more important in our body. So the primary structure of the amino acids is, you can see here, CONH amide group is attached. So the pink form is amino acid, and the blue form is a peptide bond. Peptide bond means what? CONH group is there. So see here the structure properly. CH, CO1O, NH2 is a amino acid peptide amino acids, and the bond between the amino acid is a peptide bond. So it describes the sequence of amino acids, that is a positional information in a protein. Fine. This one is a secondary structure of protein, which shows that CONH group. So C11O and then NH group in a spiral form, or we can say in a helix shape, right? Or we can say zigzag shape also of the amino acids, which contains CO, that is a carbonyl, as well as NH, amide peptide bond, CONH peptide bond. The same example of a keratin or fibrin, silk fiber, which is given the different structure of a protein in a spiral structure in a helix form. And this one is a pleated sheet, which forms a CO and NH group over here. The next one is a tertiary structure of a protein. So this structure is somewhat like a long chain of polypeptide link. Now peptide means what? CONH bond link. Poly means what? Number of peptide links. So polypeptide link is folded in many points like a coil shape. So according to the van der Waals attraction of force, the hydrogen bond, ionic bond, and the disulfide bond exists over here and can form a tertiary structure of a proteins, which we can give an example of a myoglobin protein. This is just for the information. So we can just say a tristructure of a protein consists of what? That is due to the van der Waals force of attraction, hydrogen bond, ionic bond, and the disulfide bond is there. See here the example of a tertiary, so like myoglobin spiral form, and then link is there. Enzymes. Now the next one is a quaternary structure of a protein. So the quaternary structure of protein is found in some complex proteins. That is a formation of a 2 or more polypeptide bonds. That is a alpha bond as well as beta bond. So this structure is a quaternary, and it's a hemoglobin protein. So hemoglobin is more important in our body. The number of hemoglobin shows the red corpuscles present in our body. So here there is structure of a 2 alpha as well as 2 beta chains, which is present over here. So these are all the 4 terms of protein. There is a primary, secondary, tertiary, as well as quaternary structure of the proteins. See here, you can see heme groups are there. So hemoglobin, it has 4 subunits, that is 2 alpha subunits as well as 2 beta subunits are there. Now about the nucleic acid. So every generation of each and every species resembles ancestors in many ways. So some we can say the heredity, we have studied in 10th standard about the heredity. So the heredity shows color of skin color, hair color, the nature of eyes, eyebrows, earlobes. So this you get from the heredity. So same as well as the medical symptoms also, like diabetes or changing nature. This all depends on the heredity. So heredity means what? That is, in the biological term, it shows a biomolecules called nucleic acid. So there are 2 types of nucleic acids, that is a DNA and RNA. So what is DNA? Deoxyribonucleic acid, and RNA, ribonucleic acids. So this simply terms a long chain of polymer is called nucleotides. So the number of nucleotides is called polynucleotides. Poly means number, long chains of a polynucleotides are there. So see here, there are two types of nucleic acid, DNA and RNA. So we have might be seen the DNA in any videos or pictures. So DNA shows the nature of the person to the other person, like heredity, like grandfather having the same DNA, 70% DNA to the son or the grandsons, like that. So it shows what, for example, sugar. So beta D ribose sugar is present in the RNA, and beta D2 deoxyribose sugar is present in DNA. The structure of a ribose includes what? CH2OH and then O form. There is an ether form. So there are two different things. What is the difference over here? You can see. This one is a position CH2OH, OH, H, and HOOH. See here, there is a very minor difference over here. Here it is a OH and here is a only single H is there. So very minor difference is there. So because of that, it shows beta D ribose and beta D2 deoxyribose. Now heterocyclic base. Base itself shows nitrogen is present over there. So the pyrimidine as well as purine. So pyrimidine consists of two nitrogen in the first and the 3 position with the benzene ring, and purine consists of a dicyclic compound in which it shows the, so see here 1, 2, 3, 4, 5, 6, and then 7th, which is attached with the 5th carbon, not with the 4th carbon. It's the number 7, 8, and 9. So this structure is a purine. So it's a dicyclic compound with the 1 hexane and 1 pentane group. Okay. So here adenine guanine. So ATCG is formation over here in the DNA. A means what? Adenine, G guanine, cytosine, and thymine. And the last one is uracil, that is a capital U, which is present in the nucleic acid. Nucleic acid means amino NH and then acid COOH group is there. So the DNA contains AGCT base, while RNA contains AGCU bases. So adenine, the structure all is very important over here. Adenine structure is what? Benzene with the 1 pentane, that is NH2 and NNH. Here guanine, it contains 1 ether formation with the same, like just like that, the adenine over here it is a NH2 and here replace the OH, O, cytosine with the NH, O, N and NH2 cytosine, and thymine, NH, double bond O, NH and CH3 and thymine, as well as uracil. This is quite important for the structure of the DNA. Now phosphoric acid. Phosphoric means consists of phosphate. Phosphate means PO4 minus. So the structure, it shows like P4. So how many 4? Oxygen is 4. P1. So oxygen requires 2 bonds. So O minus and O minus over here. So attached with the C5, the under sugar level over here. So see here that nucleoside is attached with the base. Phosphate is attached to the sugar and base. See here, this is a nucleotide. This is a nucleotide. So it is attached with the base as well as the phosphate. This is the sugar structure. HOH, CH2, and it is attached with the phosphate over here. So this is the nucleotide. How you will remember phosphate? That is PO4, 2 minus, and that is the third minus. It is attached with the CH2, and then nucleotide, that is sugar, and then base is there over here. Yeah. So phosphodiester linkage. So how you will remember this one? This is like a phosphate, sugar, and the base. When this reacts with each other, it forms what? Phosphodiester. What is phosphodiester? That is PO4 with the ester linkage is attached with the 2 carbon over here. See here, in short, we can do like this: base, sugar, phosphate, and then again it is repeating: base, sugar, phosphate, and then base, sugar, and then last phosphate. So this is what? Nucleic acid in a number form. So here you can see the double helix structure of a DNA, that is a deoxyribonucleic acids, in which you shows the replicate of the spiral of the DNA structure. P means for phosphate, D deoxyribose, A adenine, T thymine, C cytosine, and G guanine. See the linkage between the AT is double bond, CG is triple bond. Again, the opposite will come here, TA, right? So this is how the double helix structure will form. See, AT double bond, CG triple bond, the bond between these things. So see here, AT are joined together with a 2 hydrogen bond, and CG are joined with the 3 hydrogen bonds. There should be some different distance between the 2 bond, that is AT and CG is also there. See here, the nucleic acids. So the backbone strands of the DNA is formed by the sugar phosphate sugar chain, that is we have studied right now, sugar, phosphate, and sugar. So here adenine, AT, adenine, thymine, guanine, cytosine for the DNA, and uracil RNA is there. So the bond is 2 hydrogen bond and 3 hydrogen bond. So this is sugar and then phosphate. Now the importance of a nucleic acid. So what is the importance of a nucleic acid? As we all know that a DNA is a responsible factor for the heredity, and shows the function of a nucleic acid, which is the protein synthesis in the cell. So actually the proteins are synthesized by various RNA molecules in the cell, but the master of the synthesis of a specific protein is present in the DNA. So so from the test of the DNA and RNA, we can able to understand which person, because every person in the human being DNA and RNA is different. Thank you. Hope you understood the topic very well.